Hi everyone, my name is Gopal Sharma and I'm a second year master's student in the plant science program at UBC. I'm co-supervised by Dr. Thorsten Kniffer and Dr. Gertrude Barar, and today I will be presenting some results from my master's project. The title of my seminar is a determination of physiological stress thresholds for improved selection of drought-resistant wheat genotypes. To start, I want to give a bit of a background information on wheat and the impact of drought as well as drought strategies. Wheat is one of the most important field crops in Canada and worldwide. 32.7 million metric tons of wheat was produced in 2019 in Canada. It is not only an important food source, but it is economically important as well. Wheat is primarily grown in Western Canada, which consists of British Columbia, Alberta, Saskatchewan, and Manitoba. In this region, wheat relies on rainwater irrigation, which can be problematic as Western Canada has a history of droughts due to its topography. The combination of the reliance on rainwater for irrigation and the frequency of droughts make drought performance of wheat an important topic. There has been a lot of literature published on drought studies in wheat. Drought conditions have been found to affect wheat in several ways. For example, decreases in plant height, yield, and both above and below ground biomass have been noted. Root hydraulic conductance can also decrease due to superization, and the elongation of roots were noted to slow down with drought stress. Wheat plants can utilize different strategies to combat drought conditions, and this has been a big topic of concern, especially here in Western Canada. One of the strategies noted was the use of deep root architecture to tap into deeper soil water. Increased flagleaf wax density in drought-tolerant wheat lines has also been noticed. But the focus of my presentation today will be another strategy, which is osmotic adjustment, which has been noted to maintain yield in other studies. To better understand osmotic adjustment and the physiological stress thresholds, which I will be sharing in the results section, I would like to take some time to provide a brief background. Plants take up water through their roots and lose it through transpiration, which can be seen in the diagram here. The difference between the root water uptake and the plant transpiration determines the xylem pressure. The water molecules are connected using hydrogen bonds, creating a hydraulic continuum. The water potential of a system is the potential energy of water within a system compared to pure water. It provides a way to determine the plant water status. As soil dries, a more negative water potential is seen in the plant. Water moves to an area with a more negative water potential. The diagram on the right here shows that cells can have water movement from and to the xylem to reach an equilibrium. This also means that the water potential of a leaf would be equal to the xylem water potential, which then would be equal to the xylem pressure. The water potential is made up of two components, the solute potential and the pressure potential, also known as turgor, and this can be seen in the equation above the diagram. The solute potential makes up one part of the water potential equation. It is the amount of osmotically active ions, or solutes, present in a cell. An increase in solutes will cause a decrease in solute potential. In the diagram above, the cell on the left has fewer solutes than when compared to the cell on the right. Therefore, the cell on the right has a more negative solute potential, which also decreases the water potential of the cell. As the solute potential and the water potential of the cell on the right are more negative, the water flows into the cell from the xylem. Now, this is not the case on the cell on the left, in which case the water is flowing to the more negative area, which happens to be the xylem. This strategy of accumulating more solutes and ensuring that water is retained in the cell is called osmotic adjustment, and it helps a plant to maintain its turgor. In the plot on the right here, the lower line has a decrease in solute potential as the soil dries. This is osmotic adjustment occurring within a plant, which will then help maintain the turgor as the soil dries. The turgor is another component of the water potential calculation. It is the pressure within a cell that pushes the plasma membrane against the cell wall and can describe when a plant will wilt. In the diagram above, the xylem pressure is more negative and the water molecules are more stretched, which causes the water to flow into the xylem from the cell to reach an equilibrium. As this is happening, the turgor is decreasing and it will eventually hit zero, which will cause the turgor loss point. The plot on the right here shows turgor maintenance as the soil dries until it hits a certain threshold, at which point it starts dropping, and then it eventually hits the turgor loss point. Now, moving on to my experiment. The focus of my experiment was to determine the physiological canopy stress thresholds of 3 elite wheat cultivars with an emphasis on leaf-water relations. My hypothesis was that the physiological thresholds can be used to assess drought performance. Custom PVC pots were created, which had a depth of 60 centimeters and a diameter of 15 centimeters. This was done to prevent any restrictions on root growth. The bottom of the pot was mesh held together by a hose clamp and duct tape, and this allowed water to drain from the pot but keep the soil in. A mixture of 2/3 sand and 1/3 of peat and perlite based potting mix was used. The plants were well watered until the dry down period. 3 cultivars were used in the experiment, and these were chosen based on their drought performance. Superb was picked as the drought susceptible line, as it was noted to have not performed as well in drought conditions when compared to Stettler in a previous study. This was also the reason that Stettler was picked as the drought tolerant line. AAC Viewfield was also chosen as a drought tolerant line because it was noted to have performed well in the drought conditions in the field. Now, in terms of the treatments, once the plants hit the early anthesis stage, 2 different treatments were applied. Treatments were randomized. 12 control plants were maintained at 90% bulk soil moisture. 18 drought-stressed plants were not watered once the dry down period started. These plants can be seen in the picture here on the right. The control plants were watered every second day. The plant weight was used to rewater the plants. The dry down period was 2 weeks long, at the end of which the drought-stressed plants were very dry. Several physiological measurements were taken during the dry down period. The water potential was measured using a pressure chamber, which can be seen in the top right picture. The pre-dawn water potential was taken in the morning on plants covered with black bags to prevent transpiration. The midday water potential was taken around noon. Either the flag leaf or the penultimate leaves were used for these measurements, and the leaves were kept on ice after the excision until they were placed in the pressure chamber. We used a stereoscope to get a closer look at the leaf, and you can see how the leaf looked under the stereoscope as the pressure was being applied. And the picture to the left of the water potential pressure chamber. An osmometer was used to find the solute potential of the leaf samples, and this can be seen in the bottom left picture. A small leaf sample was kept in the -80 freezer before being taken out and centrifuged. 10 microliters of the osmotic sap were then placed in the osmometer and measured the solute potential of the sap. The plant weight was obtained using a high-resolution scale. The weight gave the bulk soil moisture and daily transpiration rate of the plant. The bulk soil moisture values were calculated by taking soil samples and finding the saturated and the dry weight of the soil. The difference between these two amounts was the amount of water that could be held within the soil. Before the pots were seeded, the saturated or 100% soil moisture weight of each pot was found. Using the 100% soil moisture pot weight and the water weight of the soil samples, the 90% bulk soil moisture could be calculated. The transpiration rate was calculated by finding the difference in plant weight between days. Leaf relative water content was measured by subtracting the fresh weight from the dry weight and dividing by the difference between the saturated and the dry weight of the same leaf sample size. And this provided us with an estimation of the leaf water status. In addition to these measures, pictures were taken daily against a white background which helped to track plant growth. They were taken on a platform, imaging platform, which can be seen on the bottom left picture, in which the plots were placed on the cart, and a camera was kept a certain distance and a set height, and the white background had a scale in the form of tape, so that we could then compare plants between days and between cultivars. Now, the most exciting part, my results. I have provided a table here with the soil moisture thresholds and the standard errors for each physiological measurement. We looked at the relative water content pre-dawn and midday leaf water potential, as well as the solute potential. The data from these measurements were put into a piecewise linear regression to identify breakpoints or thresholds. Basically, what a piecewise linear regression does is take the dataset and determines the point at which there is a change in the slope. These thresholds are indications of physiological changes in a plant, and they represent the soil moisture percentage at which there is a shift in the slope or a drop in the physiological measurements. They provide information on drought-induced changes to the water relations in wheat. As seen in the table, all 3 lines have different thresholds. Superb has a threshold of approximately 36%, and Stettler has one around 47%, and this is across all 4 physiological measurements. For AAC Viewfield, it acted differently compared to the other 2 lines. The solute potential threshold occurs at 59%, followed shortly by the relative water content and the midday water potential thresholds. The final threshold, which is the pre-dawn water potential threshold, is passed at 43% bulk soil moisture. Looking at the standard errors, the thresholds have very little overlap. Furthermore, it appears that AAC Viewfield drops in solute potential early on as the soil dries. This is osmotic adjustment occurring and the plant's way of actively regulating itself to survive drier conditions. This is not seen in Stettler and Superb, where the plant only goes up to a certain threshold before all the physiological measurements crash. Let's take a closer look at these thresholds. Now the question might be, what does this all mean in terms of the plant? Here I have plotted the physiological thresholds on a bulk soil moisture scale, and I've also included some pictures before and after the thresholds were passed. So green is the relative water content threshold, red is the pre-dawn, and blue is the midday water potential threshold, and finally yellow is the solute potential threshold. Starting off with the range, as you can see, both Superb and Stettler have a very narrow range for the thresholds. They all occur very close to each other, and that can be seen in this diagram very clearly. AC Viewfield, on the other hand, has a much larger range, and while it hits the solute potential, relative water content, and midday water potential thresholds first, its pre-dawn water potential does not crash until all of Stettler's thresholds have been passed. I have also included pictures of the plants before and after these thresholds were passed. Interestingly, all three cultivars looked healthy prior to the thresholds being passed, but once they did, they all looked dry, especially at the base. Another interesting thing was that Superb and Stettler had a very narrow range. It was seen that as soon as those thresholds were passed, the plants became very dry. In contrast, AAC Viewfield did not show any visual signs of drying until the pre-dawn water potential threshold was passed. This could be in part due to osmotic adjustment occurring. Now, in conclusion, these results show that AAC Viewfield uses osmotic adjustment, which is seen by the drop in solute potential first. This strategy helps cells retain water and leads to better performance in dry conditions. Furthermore, soil moisture thresholds can be practical tools in the field. They can provide an alternate way to assess drought performance, which can be used to compare different genotypes. They can also be used for precision irrigation, which can then be used to conserve water. I would also like to take the time to mention that we are currently working on a manuscript based on this data, and the data should be available later this year for those of you who would like a closer look. To end off, I would like to take the time to thank my supervisors, Dr. Thorsten Kniffer and Dr. Gertrude Barar, for their constant advice during this experiment. I would also like to thank the UBC greenhouse staff and my fellow lab members in the crop pathology and genetics lab, as well as the Kniffer plant physiology lab, for their help. And finally, thank you to everyone for watching. I look forward to discussing these results with you. Thank you.